Hello, and welcome to the December Mission Impact Volunteer Call on Disease-Modifying Therapies. Today's presenters are Joshua Veith, PhD, and Stephen Karpin, PharmD.
Thank you for the introduction, Brian, and thank you to all of you for joining us for today's Mission Impact Volunteer Call. I'm Josh Veith, and I'm on the research team here at Breakthrough T1D, and today I'm going to be discussing some updates from the disease-modifying therapy portfolio. You have all seen a slide like this before showing the staging of type 1 diabetes. And this staging really is involved with our strategy as our primary research areas being early detection, focused on those before or at stage 1, disease-modifying therapies, which really span those in stage 1, 2, or 3, and beyond if we talk about combining those with cell therapies, which we'll discuss in a moment. And then finally, our cell replacement therapies portfolio and our treatments portfolio, which are focused on those living with established type 1 diabetes in stage 4. The goal of disease-modifying therapies is to reverse the course of disease. So disease-modifying therapies are therapies that change the course of disease aimed at people at every age and every stage of type 1 diabetes. And really what we're trying to do here is to prevent, halt, or reverse the loss of those functional insulin-producing beta cells. Now, how do we do this? Really two ways. We want to rebalance the immune system and restore the balance in the autoreactivity that's occurred. And we also want to preserve or regrow functional beta cells. Now, there are several DMTs that have been supported by Breakthrough T1D in clinical trials around the world, and one that has been approved by the FDA for those in stage 2. To start, we're going to review a little bit about T1D biology. It's important to always remember that type 1 diabetes is an autoimmune disease. And when we talk about immune-related diseases, we want to remember that the primary role of the immune system is to determine self from non-self. In its simplest form, the cells of the immune system have a job to recognize threats and then respond accordingly. Now, you want to maintain a balance so that the immune system is active enough or providing enough surveillance and recognition to be able to identify and eliminate threats. But you don't want it so active that it starts to recognize your own cells or parts of your own cells as a threat and respond accordingly. So when we have an inability for the immune system to properly determine self from non-self, this can lead to the development of autoimmune diseases such as type 1 diabetes. And it's important to remember that the underlying breakdown and regulation across autoimmune diseases is similar, and we're investigating ways and the commonalities between these, and ultimately the target dictates what type of autoimmune disease we're talking about. Now, if we dig a little bit more into the stages of T1D progression, our strategy really focuses on the biology behind it. At the very earliest stages, there is a genetic susceptibility. We know that there are a list of 30 plus genes that are associated with a risk of developing type 1 diabetes. But on top of this genetic susceptibility, you also have to have an immune activation event, some event that causes the immune system to recognize the beta cells as a threat. Now, when this happens, when the immune system activates, one of the first signs of this is the presence of autoantibodies. And you can think of autoantibodies or antibodies in general as a flag that's sort of thrown by the immune system to mark something as a threat. You can think about it either as a football ref throwing a flag on a play, or maybe a more apt comparison would be when you have a park ranger that goes through a forest and sort of spray paints a tree, and that marks that tree to be cut down. That's really how antibodies work within the immune system. So what this means is that those presence of those autoantibodies means that that immune reaction to the beta cells has already begun. And the start of type 1 diabetes is prior to what we measure as stage 1. It happens far earlier than diagnosis, where you have symptoms develop. So again, in stage 1, we have the presence of multiple autoantibodies, and this is just a sign that that reaction against the immune system has begun. Now in stage 2, the immune system has started to go after and attack some of the insulin-producing beta cells. This means that there's still those autoantibodies present, But now you lose enough beta cells where you start to have some abnormal blood sugar readings. Now, at diagnosis, you've lost enough of those insulin producing beta cells to have symptoms occur and to require exogenous or injected insulin. You're still producing a little bit of your own insulin, but not enough to meet your needs. And finally, as you can see, the beta cells continue to deplete, and the beta cells are these tan cells here in the islets. In established type 1 diabetes, you've lost those insulin producing beta cells. This is what we call established T1D. So you're completely reliant on exogenous or injected insulin. Now, how do we focus on this biology of disease and disease modifying therapies with regard to our priority areas of our strategies? And really, there's two sides of disease modifying therapies. There are immune therapies and beta cell therapies. On the immune therapy side, we talk about how can we rebalance the immune system, and we really target this in three different ways. One, we want to disable the autoreactivity. So we want to go after the cells that are driving disease. We talk about these as T cells or B cells. And these are cells in the immune system that are sort of the soldiers that go out and go after the threat. So some of the therapies, such as teplizumab, go after T cells, which are driving the destruction in the cells that are actively going after the insulin-producing beta cells. So part of our therapeutic approach is to use different drugs and antibodies that can deplete or diminish the activity of these autoreactive cells. Secondly, we want to enhance regulation. As part of the balance of our immune system, there are natural built-in mechanisms by which the immune system can regulate itself. And part of the dysfunction in autoimmune diseases such as type 1 diabetes is that this regulation is diminished or not happening or as strongly as it should. Therefore, the second part of our immune therapy strategy is to enhance the natural regulation of the immune system. This means bolstering the activity of what are called T regulatory cells. So pushing those cells to produce more cytokines or chemicals that calm down the immune system, increasing the number of those T regulatory cells and regulatory cells in general. And also that we can use tolerizing approaches to enhance regulation. So we can show little bits of the beta cells or the proteins that are causing or activating the immune system in a way that teaches the immune system to stop responding to it and to quiet down that autoreactivity. Finally, with any immune response, you have inflammation. Inflammation is the result of the chemicals that the immune cells use to talk to each other. And this leads to tissue damage and inflammation within the tissue. We talk about insulitis in the pancreas. So the third prong of the immune therapies is to how to disrupt that inflammation and stop it from causing more beta cell damage and therefore more of an immune reaction. On the other side, we have beta cell therapies. And the main question here is how do we protect or expand functional beta cells? So under reducing beta cell stress, we focus on how to protect them. How can we diminish the effects of that immune activity on beta cells, which causes stress, to stop that stress from occurring, allowing those beta cells to continue producing insulin at normal levels, and to stop them under those stress conditions from showing flags or messages to the immune system that increases the attack. And finally, we talk about beta cell regeneration. In the context of disease-modifying therapies, using drugs that can specifically target beta cells to divide and produce more of themselves to replenish those lost beta cells is a goal and part of our strategy. Where are we today? We've talked about this before, but it's worth repeating that we are the first disease-modifying therapy for type 1 diabetes, Tzield, and it was approved by the FDA in November of 2022. This is a huge win for the field, and it paves the way for many other therapies and many of the other approaches that we're currently moving through clinical trials. There are several promising disease-modifying therapies that have shown an ability to slow or halt the progression of type 1 diabetes, either by resetting the immune system or preserving beta cell function in early clinical testing. And finally, we've shown that several therapies that can induce beta cell regeneration, and we're working on how to target those in a safe way to get them to the cells where they need to be and to be able to develop therapeutics that work for individuals with type 1 diabetes. Now, please don't go into too much detail on this slide in terms of all of the different immune pathways. I'm not showing it to confuse anybody. I know it's a lot of information. I'm really showing it because I want to make the point that we know now that we can change the course of type 1 diabetes progression. If you look at all of the cells in this map, these are all sort of a simplified version of the pathways that are involved in the pathogenesis or the progression of type 1 diabetes. Those that have sort of red lines or arrows are involved in the progression of disease. And those that have green lines or arrows would be more involved in stopping or halting disease or reversing disease in certain cases. Now, on this picture graph, each of the drugs represented, see rituximab, imatinib, teplizumab, each of these has shown efficacy in delaying or halting the progression of disease in individuals with new onset in phase 2 trials. There's a couple things I want to point out. One, there's a large number of them that have shown efficacy. We have a number of shots on goal here that have the potential to develop into therapeutic strategies that can help patients. The other thing I want to point out is about our strategy. You can see that these all hit very different portions of the immune processes or the biological processes involved in the immune response to beta cells. And the goal here is to utilize different what we call mechanisms of action. And the mechanism of action of a drug is how it works. So we talk about rituximab is able to stop the B cells from doing what they're doing. Teplizumab causes the T cells to become exhausted and stop attacking the beta cells. And you can see these mechanisms of actions are sort of spread across what we call the immune map, meaning that they go after different parts of the imbalance that has occurred. This is really important for strategy because in each of these trials, some patients responded and some didn't. And one of the focuses we have now is to figuring out why some patients respond to one drug versus another, and also to improve the overall efficacy by designing strategies to combine these in different ways, to combine something that may work in the B cell area with something that works in the T cell area to develop a more potent and a more effective combination therapeutic. I also want to point out that there's some areas where you don't see drugs, in the Treg space here, in the beta cell regeneration space. We are developing areas or strategies in these areas, but they haven't yet progressed to phase 2 trials. There's a number of strategies for how to bolster Tregs and the work they're doing. We also have a number of shots on goal in the beta cell regeneration space, and our goal is to continually move these through the pipeline and move them into phase 1 and then phase 2 trials, and eventually into safe therapeutics for patients. And that speaks a little bit to where we're going. So what are we talking about when we talk about disease-modifying therapies for tomorrow? Our vision is to make commercially available disease-modifying therapies that slow, halt, or reverse the progression of type 1 diabetes available to people of all stages and all ages of type 1 diabetes. How do we get there? We really have a three-pronged approach. First, we want to build on the momentum of recent clinical successes. This includes the approval of teplizumab for stage 2 type 1 diabetes, but also all of those phase 2 trials I just showed you that show that we can either support beta cells or rebalance the immune system using these therapies, using these drugs, and to move those further through the pipeline. We want to continually build on the momentum of these successes. We want to clear a path for the regulatory approval of these once they show efficacy. And in the next part of this MIB, Stephen's going to talk a little bit about some of the regulatory work we're doing around C-peptide that's very much so involved in that part of the strategy. And finally, we want to look at how we can use combinations and therapies that can address all ages and all stages of disease. So in order to talk through some of the ways we're moving that strategy forward, I want to highlight a few projects where we're moving the needle. The first project I want to talk about today is a project from Dirk Homan at Mount Sinai here in New York. And the goal here is to identify markers of T1D progression. And it's a coordinated effort that's being funded with a combined almost $1.9 million investment by Breakthrough T1D and the Helmsley Charitable Trust. And that's adding to $3.7 million in funding from the NIH, specifically from NIDDK, whose funds come from the Special Diabetes Program, which Breakthrough T1D advocates for each year. One of the most critical gaps in speeding up and creating more efficient clinical trials is identifying biomarkers or factors from patients that are linked to T1D progression. And the reason this is necessary is we have to readily and quickly be able to determine whether somebody is responding to a therapy or progressing through disease. And this project seeks to overcome that by taking a coordinated effort that requires multiple funding agencies and laboratories all across the United States to do this work, to do a deep dive into these samples, to identify markers that can help with speeding up and improving the clinical process. Now, it's focusing on using serum because it's very easily both collected and stored from patients. And the goal here is really to build the most comprehensive database of T1D, what's called pathogenesis, or factors associated with the progression of disease ever compiled. It includes a number of important factors, including one of the largest repositories of samples ever collected. It includes, like I said, experts from across the field at leading institutions across the United States. And ultimately, it's going to use artificial intelligence and machine learning to identify the new biomarkers and then to create a publicly available database that anybody's going to be able to go into and mine the data and take their unique perspectives to find factors and biomarkers in this data set. The next project I'm going to talk about comes from Dr. Samuel Stevens at the University of Iowa, and this is a project that was just funded. And the goal here is to unlock the mechanisms behind beta cell stress. As we talked, a number of slides back, one of the goals of the disease-modifying therapy program is to help support beta cells that are under immune attack and to stop the cellular stress that causes reduced function and ultimately even more immune reactivity from the immune system. Now, we already know that the cytokines, they're inflammatory signals that come from the immune system, can change the way the beta cells work, can reduce the amount of insulin they produce, and cause more recognition and response from the immune system. If we can identify the mechanisms that are responsible for how this stress changes the way beta cells function, then we can identify some novel therapeutic targets that may be able to support beta cell function when there's an autoimmune attack. And we can also look for novel markers for assessing the beta cell health under stress conditions. Now, the goal of this research is to identify how exposure to these immune cytokines changes the way proteins are processed by the beta cells, how the beta cells themselves actually do their job, and also identify how this changes what factors or proteins are displayed on the surface of the beta cell that the immune system can see. Now, this is going to be done using human cells, but in the context of in petri dishes, right? They're going to treat these in the laboratory and look at the changes that happen in these cells. Importantly, the third part of this work is going to validate these findings. So any changes they observe, they're going to use samples from the NPOD repository to identify and validate these to show that they are present in individuals with type 1 diabetes and to validate them as potential targets for a therapeutic approach. The next project I'm going to talk about comes from Inspiro. Now, this is an industry partnership that we're involved in. Inspiro is based in Switzerland, and they're also trying to unlock the mechanisms behind beta cell stress. And I'm highlighting this to show that we're going after this problem in multiple ways. Now, there's in the body, there's a certain amount of communication that happens between cells, including between beta cells and the immune system. And this is really at the heart of the immune reactivity of the immune cells to the beta cells. And beta cells express a number of proteins or factors on the surface that can either activate, and these are called HLA class 1 proteins, as well as repress or calm down. And we call these PDL1, the immune attack. What's missing from the field is really a high throughput way to determine how drugs can reduce or increase the expression of these different factors on the beta cell surface. Now, Inspiro has developed what's called a microtissue technology. They're able to take human islets, dissociate them into single cells, and then put them together into tiny little microtissue islets. So basically, you have a plate with 300 some wells on the plate, and each of these wells is a little mini islet that can be representative of what happens in a human islet in the pancreas. And they're using this process to validate therapeutics that are on the market for their ability to increase or decrease these immune communication proteins on the surface of the beta cells. And what they're trying to do is to develop a platform that can rapidly assess whether or not a drug can change the way the immune system and beta cells are interacting in order to reduce the amount of stress on the beta cells. And the goal of this is really to eventually utilize this platform to investigate novel or new therapeutics that can be used for further clinical development. So this is an investment in the field and in a technology that can benefit strategies across the disease-modifying therapies portfolio. Now, one of the things I mentioned as a goal for our disease-modifying therapies is the development of combination therapies. And one of the combination therapy clinical trials we just funded goes to Dr. Lorenzo Piemonte at the University of San Rafael. Now, as I said before, using combinations of therapies that target different parts of the immune map really gives us an ability to look for strategies that can increase the efficacy of the approach, as well as the duration of response and the number of people that are likely to respond. Dr. Piemonte is going to run a pilot clinical trial that's really testing a combination that uses anti-thymocyte globulin, and you might have heard this. It's called ATG, and this is shown to be effective in those in new onset. It depletes the T cells and may also encourage the development of Tregs. Now, what he's doing is he's adding to this a drug called latorixin. And what latorixin does is it blocks the recruitment of some of the cells that cause the inflammation. And one of these cell types are neutrophils. And really the goal here is determine whether you can add latorixin and whether or not patients receiving both drugs perform better and have longer lasting beta cell function than those that receive ATG alone. So the goal here is to evaluate the efficacy of using sort of a short dose of ATG with a longer treatment of latorixin, and to see how this can halt the autoimmune destruction in those with new onset. We're very excited to see this project move forward. The last update I want to give is actually an update from something we talked about during the last disease-modifying therapy presentation. And this is Diagnode 3, which is a phase 3 trial being run by Diamyd Medical out of Sweden. And you'll remember that this is testing 3 interlymphatic injections of GAD65 in patients with new onset type 1 diabetes. They've had a number of recent and pretty incredible updates, and I wanted to share those with you today. The first is that their recruitment is progressing extremely well. They're open in countries throughout Europe and now in the US. They've recruited 180 out of 330 subjects as of this month. And importantly, there's been a 0% dropout rate, meaning this therapy has been both well received and well tolerated by patients. In February earlier this year, the FDA granted fast track designation for Diamyd in stage 3 type 1 diabetes, and then in July granted fast track designation for Diamyd in stages 1 and 2. And what fast track designation means is it allows a therapy that addresses an unmet need to get to patients faster by improving the efficiency of the review process. This means that the company gets more frequent and often interactions with the FDA. It's a rolling review process, which means that they're able to turn in portions of the application approval over time versus in just one application. And it opens them up for accelerated approval if certain conditions are met. Now, they had an internal analysis at 6 months in July, and it was recommended that this trial continue, meaning that it was shown that the trial is likely to answer the question of whether or not this therapy is effective for new onset type 1 diabetes. And importantly, they met with the FDA, and they are going to do an additional interim efficacy readout early next year at this time, to prepare for what's called an accelerated approval to the FDA. And what the purpose of accelerated approval is to make drugs that fill an unmet need and show efficacy in ongoing trials available for patients sooner. It allows them to use a surrogate endpoint such as C-peptide, and the FDA stated that they were open to using C-peptide as a surrogate in this case, which is important as this is something that Breakthrough T1D has been advocating for and working for a long time. And the ultimate goal here is to shorten the timeline to approval and quicken the availability to patients. And we're very excited to see this interim efficacy data readout and for this to potentially be considered for accelerated approval. Now, just a quick update on where we stand in the disease-modifying therapies portfolio. If we look at where disease-modifying therapies are within the entirety of the research active grants, it represents about 30% of the portfolio, a total of current investment of $130 million. And as you can also see, this goes from early discovery research all the way to late clinical trials such as DIAMID. And we're continuing to work at moving these therapies and these different approaches from the discovery phase of research into early preclinical, late preclinical, and then clinical testing to take the best opportunities to help those at all ages and all stages of disease and to turn them into therapies and to get the clinical data necessary to make them available to patients. And with that focus, I also want to give an overview of where we stand for the clinical trials we're currently supporting under the disease-modifying therapy program. And you'll see that we're currently supporting 12 different therapies at various phases of development. I talked about the DIAMID Diagno3 trial, which is in phase 3 development, as is an extension of the verapamil trial. We have a couple ustekinumab and then a combination trial of teplizumab and verapamil in phase 2, or late phase 2, early phase 3, a large number in phase 2, and we've recently launched a trial in denosumab, which is a phase 1 proof of concept trial, looking at a drug that's normally used to treat osteoporosis and bone cancer that also has the ability to reset the immune system and may have efficacy in type 1 diabetes. So as you can see, we're continuing to look at all the different, what we spoke about earlier as mechanisms of action and advancing therapies across that immune map to both rebalance the immune system and to support and preserve beta cell function in those with type 1 diabetes. And now I'm happy to turn things over to my colleague, Stephen Karpin, who's going to talk a little bit more about one of the priorities I brought up, which is regulatory approval of these therapies. And he's specifically going to talk about our work around C-peptide and some of the work we're doing with regard to scientific and regulatory considerations.
Hi, everybody. I'm Stephen Karpin. I'm the Senior Director on the Regulatory Affairs team here at Breakthrough T1D, and I'm going to talk to you today a little bit about some of the scientific and regulatory considerations for C-peptide as we think about developing new disease-modifying therapies. The Regulatory Affairs team is very active in working towards advancing the regulatory status of C-peptide. We do quite a bit of work, including looking at clinical trial data, both existing data, how that can help with the status of C-peptide, but also thinking about new trials that can generate data that we don't yet have. We work very closely with the regulators and with industry in terms of the regulatory pathways that are available, how we can improve those pathways, and what industry-sponsored clinical trials could look like to help support C-peptide. We also do a lot of consensus building with the research community, getting a lot of publications out there, really making the case for why C-peptide is a validated surrogate endpoint, why the regulators should see it as such, really helping to make that a much more available tool for industry sponsors to use. When it comes to engaging the regulators, we do this in a number of different ways. We have quite a few informal scientific discussions with them. Sometimes this looks like sharing new publications with them, but making sure that their knowledge on the topic is as up to date as possible. We also meet regularly with agency leaders, including the commissioner, to make sure type 1 diabetes is front and center on their minds and that they're working just as hard as they can to improve the regulatory pathways in type 1 diabetes. And then we do some more technical work with the regulators, like submitting specific data or new tools that can be used as part of the regulatory decision-making process. We work closely with industry partners throughout the entire drug development cycle and pipeline. Sometimes this means giving them some advice on better designs or improved designs for their clinical studies. Sometimes this means reviewing the materials that they're getting ready to submit with regulators, helping to give them some guidance on their overall regulatory strategies. But a key part of our regulatory strategy is making sure that all of the sponsors in the type 1 space are as empowered as possible to make as much progress as they can, as quickly as they can. So let's talk a little bit about why disease-modifying therapies is a new approach for how we think about managing type 1 and why that means we need to think about new endpoints in our clinical trials. Historically, when we think about treating the insulin deficiency component of type 1, we're really thinking about glucose control, typically through insulin. And the way we assess that in clinical trials is kind of what we do now: A1C, hypoglycemia, how much insulin you're using. But the main focus here is managing blood glucose. When we think about disease-modifying therapies, we're not really directly targeting that glucose control. What we're thinking about is the underlying autoimmune disease, the attack on those beta cells. The therapies that we're developing, the goal is to slow or halt the disease progression to preserve as much beta cell function as possible. And so when we think about clinical trials with those therapies, rather than measuring A1C or hypoglycemia, the best thing we can look at is directly looking at beta cell function. And typically we do that through C-peptide. So what is C-peptide? C-peptide is a byproduct of insulin production in the beta cells. It is made in a 1-to-1 ratio with insulin. So when the beta cells release insulin, they release 1 molecule of insulin, 1 molecule of C-peptide. So we know it's a direct marker of how much insulin is being produced. It's also a lot easier for us to measure in the body. That gives us some benefits in terms of assessing C-peptide in clinical trials versus assessing insulin directly in clinical trials, which is a little bit less feasible for us to do. As I said, there are some challenges with measuring insulin directly to assess beta cell function. Most importantly, people with new onset type 1 diabetes already are using insulin. They have some from their beta cells, but not enough. And so they need to supplement that with exogenous insulin. When we measure insulin, we're not able to tell the difference between exogenous insulin or insulin that came directly from the beta cells. And that makes it really tricky in the context of a trial to understand, is there more insulin there because there's more beta cells, or is there more insulin because of how much insulin that individual person is using? With C-peptide, we don't have that problem. C-peptide isn't a part of exogenous insulin formulations. And so when we measure insulin in the body, even in people who are already using insulin, we know that that C-peptide is only coming from functional beta cells. And that gives us a much clearer picture of how a particular drug is affecting beta cell function in a trial. There's universal agreement that C-peptide is the appropriate marker of beta cell function. And the community generally agrees that in clinical trials of therapies that are trying to improve and preserve beta cell function, that makes C-peptide the right marker for those sorts of trials. We at Breakthrough have done quite a bit of work over many, many years to gather that consensus, to publish that consensus, to make sure that everybody does agree that there are better ways to look at beta cell function in trials than we currently do now. C-peptide being the one that we've gotten the community to rally around. And even the regulators have stated in some of their regulatory documents that they agree. You're thinking about a disease-modifying therapy that's intending to prevent loss of beta cell function. C-peptide is the right way to assess that drug's effect. And it's a big step for everyone in the community. However, there is some disagreement between the regulators and the rest of the research community about whether or not C-peptide is acceptable as an endpoint in a clinical trial. It's a good marker of beta cell function, but that doesn't necessarily mean that there's agreement that it's how we should base approval of new disease-modifying therapies. When we think about efficacy endpoints in trials, that's what we're looking at to understand a drug's effect on the body. For regulators, when they're making approval decisions, they're thinking about, do the benefits of this drug outweigh the risks of this drug? And they define benefit as anything that has a positive effect on how an individual feels, functions, or survives. Our efficacy endpoints are what we use in clinical trials to assess feels, functions, or survives. And there's two different kinds of endpoints that we use to do this. One is a clinical endpoint, and that's an endpoint that directly measures clinical benefit, how a patient is feeling, functioning, or surviving. Clinical endpoints, things like diabetic ketoacidosis, severe hypoglycemia, long-term complications, these are things that are directly impacting clinical benefit for patients. A surrogate endpoint is a little bit different. A surrogate endpoint doesn't directly measure feels, functions, or survive, but it's a different measure that predicts whether or not a drug will affect how a patient feels, functions, or survives. HbA1c is sort of our classic surrogate endpoint in type 1 diabetes. It's one that's been used for decades, and it's well established that when you have lower A1c, you also have lower risk of those direct clinical endpoints, things like microvascular complications in the long term. It gives us a lot of benefit to use a surrogate endpoint in a clinical trial. It's not always ideal to measure a surrogate rather than directly measuring the clinical endpoint. But in cases like HbA1c, where the actual clinical event might take decades before you really see that, we can't do clinical trials for decades. And so instead, we're able to use this surrogate that we know predicts that long-term complication. And that allows us to do much more feasible trials. In general, when a surrogate can be measured sooner, more easily, less invasively, or even less expensively, it has benefit over directly measuring the clinical endpoint. You can see in the picture here some of the sort of alternatives to looking at HbA1c when it comes to, you can see in the figure here some of the alternatives to measuring a clinical benefit directly when we think about disease-modifying therapies. We could do something that's a little more invasive, like directly go in, pull out cells from the pancreas, put them under a microscope and just really directly capture beta cell function. But that's kind of expensive. It's not particularly good for the pancreas or the beta cells that are there. It's invasive. And we can think about things like C-peptide as a surrogate. We can measure this through a simple blood draw. It's easy to assess. It's easy to standardize. And we get a very clear picture of what's happening in the pancreas without having to do something that's more invasive and more risky to measure that. As I said, the regulators at the moment have sort of mixed feelings about whether C-peptide is acceptable as that true endpoint in clinical trials for drug approvals. We generally think about three different kinds of surrogate endpoints, depending on how much evidence exists to support their use. With the least amount of evidence, we think about a candidate surrogate endpoint. Maybe there's something here, maybe there's a relationship that we've seen, but we really don't know enough about it to make much more of a conclusion than maybe there's something there. As more evidence accumulates that surrogate predicts a true clinical benefit, it becomes what we call a reasonably likely surrogate. We would say that surrogate is reasonably likely to predict clinical benefit. And from a regulatory perspective, at the FDA, a reasonably likely surrogate endpoint can support accelerated approval. That's approval where a company is able to market their drug, but to confirm that benefit, to make sure that it's not just likely to have benefit, but it in fact does have benefit, there's certain post-marketing requirements that the company must do to confirm benefit after accelerated approval. As more evidence accumulates, we might consider that surrogate endpoint to be a validated surrogate. That means that there's no debate. It is well established and well known that if you improve the surrogate endpoint, you will have clinical benefit. And for validated surrogate endpoints, you can get just full regular standard approval of your drug. No post-marketing requirements necessary. To move up this channel, it takes quite a bit of evidence. It's a pretty complicated, pretty rigorous path. There have been some cases in the past where we thought something was a surrogate, drugs got approved on it, and then it turns out that it actually wasn't a very good surrogate and that drug didn't have any benefit. To avoid those sorts of situations, the regulators have a very, very high bar for considering something to be a validated surrogate that can support traditional approval. At the FDA, right now, they consider C-peptide to be reasonably likely. That means that it can support accelerated approval. In Europe, at the European Medicines Agency, or the EMA, they don't use these same three designations. They just generally say either it's validated or it's not validated. If it's validated, it can support drug approval. If it's unvalidated, it could support the equivalent over in Europe of accelerated approval, which they call conditional marketing authorization. But unfortunately, in the case of type 1, they've said that conditional marketing for several reasons may not be available to our community. Where that leaves us with C-peptide right now, in the US, you can get accelerated approval based on C-peptide and then need to do some confirmatory studies to confirm that benefit. In Europe, you can't. You have to show with a different endpoint that isn't C-peptide benefit for your drug in order to get full marketing authorization. So to move C-peptide from a reasonably likely surrogate endpoint, or in the case of EMA, an unvalidated surrogate endpoint, all the way up to a validated endpoint, we at Breakthrough have put in quite a bit of effort into generating any gaps in the evidence that exist. One of those big efforts that we published was called the Trial Outcome Markers in Type 1 Diabetes Project, or the TOMI T1D project. And that study published its first results in November of 2023. That study took about 24 relatively small clinical trials. It took all of the data from those studies, put it together into one large database that had several thousand subjects. And then it used that data from all those trials together to really look at the relationship between C-peptide and the clinically meaningful outcomes that we think it predicts. This type of data is one that the regulators tend to put quite a lot of weight on when they're reviewing the evidence surrounding a surrogate endpoint. And so this was a pretty profound study for us to be able to support and to drive. The published results showed that there is a well-defined relationship between improving C-peptide and improving HbA1c. And that now is out there in front of the regulators for them to consider as they think about C-peptide. We've also at Breakthrough led publications on general consensus about the meaningfulness of C-peptide. This paper here, published in June of this year in Diabetes, summarized evidence from a number of different sources, from a number of different types of studies, including the TOMI database, including some looks to the islet cell therapy registries, and some longitudinal observational studies as well, to, again, show that when you look at the preponderance of data that exists, the community agrees that C-peptide is clinically meaningful and should be considered a validated surrogate that can support product approval at both FDA and EMA. How do we actually do that? How do we take the data we've generated, the studies we've funded, and actually put it in front of the regulators to change the way they're currently doing things and to improve the drug development process? Generally speaking, there's 3 different ways that we think about moving that regulatory needle. There are formal regulatory endorsement pathways that exist. For example, FDA's biomarker qualification program. EMA has a similar program as well. This is sort of a formalized procedure to review the data around a particular biomarker rather than a particular drug. There's the regular drug approval process, as an individual sponsor goes and talks to the agency about their specific drug. This is all data that they can take to the agency to help the case that they're making for why C-peptide should be used as the endpoint in their clinical trials. And then there's a squishy kind of third option called scientific community census, where at a certain point, C-peptide or any other marker is just so broadly used, it's so widely used, the community just generally accepts that this is the right thing to do, and the agency sort of has to follow along with that. That's part of why we tend to publish all these different consensus papers on the evidence supporting C-peptide, because the more the scientific community agrees that this is the way to go, the harder and harder it is for the FDA to not consider that as they're making their regulatory decisions. These 3 pathways happen in parallel at the same time, so we're engaging with all of them, again, with the researchers, with the regulators, with the industry, in formal and in informal pathways as we go, continuing to really beat the drum for why we need C-peptide and why the regulators should consider it to be a validated surrogate. So thank you all so much. I'm looking forward to seeing you next week and to answering any questions you may have.